before I start my professional introduction, I've just got to say I love that we start getting likes from the Zoo Facebook audience before we even begin. You don't even know what's happening and you're already excited, and I love it. So, my name is Jesse. I'm here with Exploring by the Seat of Your Pants. And for those of you who might be joining us for the first time, like a lot of our Zoo, YouTube, and Facebook audience, we are all about bringing conservation, adventure, and science into classrooms around the world. And everything we do is going to stay online forever. So if you can't catch it all, you want to watch it later, it'll all be right there for you to do that. Now, I'm particularly excited of all days you could be joining us that you're joining us today, because today it's a little bittersweet. We are wrapping up a really epic series that we've been doing in collaboration with our friends at the Toronto Zoo. So I encourage you, check out all 5 broadcasts. It's been a wild ride these last 5 weeks. As I said, they're all up on our YouTube channel, but we have loved partnering with the zoo for many years. And so we wanted to do this really special series highlighting particularly the science and conservation and collaborations that their team does to help save species at the zoo and around the world. So we've covered everything from snakes to tigers and beyond. It's been a really wild ride, and I hope you take the chance to watch them all. But today, we are going with a very iconic Canadian species with caribou, and we're going to talk about biobanking. What on earth is biobanking? You're going to know a lot more about it by the end of today's program. So without further ado, you're not here for me. I'm going to turn you over to Mary Ellen to kick us off. Thank you so much for joining us for the 5th of 5 programs, and take us away.
Hello, everyone. Thank you so much, Jesse. I cannot believe it is the end of our 5-part series here. But if you're sad like we are that it's ending, do not fret. We actually have a few more daily dives coming out over the next couple of days and weeks as well that dig deeper into more of our amazing science content and creation that we do here at the Toronto Zoo. All of this incredible research that's maybe a little bit more behind the scenes that you wouldn't normally see, or things that we don't maybe promote on a daily basis. So there's some incredible teams, amazing people that you're going to learn about, and the incredible science that they're doing. And without further ado, I'm going to turn it over to our friends out on site right now to go meet some of our in-person caribou. But before we do that, I'm going to have Jesse bring up our land acknowledgement for us. So before we get started, we would just like to acknowledge that the land that we are standing on is the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nation, Inuit, and Métis peoples. We would also like to acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit and the Williams Treaty signed with multiple Mississauga and Chippewa bands. Thank you so much, Jesse. And without further ado, let's head out on location to a very snowy Toronto. Welcome in, Jennifer, and you can take it away.
Hi everyone, I'm Jennifer. I'm the Director of Indigenous Relations at your Toronto Zoo. Greetings from our beautiful snowy caribou habitat. We are standing in front of 3 of our resident caribou here at the Toronto Zoo. So at your Toronto Zoo, our mission is to connect people, animals, conservation science, and traditional knowledge to fight extinction. So today we'd like to share how we are building stronger relationships with First Nations and Indigenous partners to help support species at risk. So how are we doing this? Well, locally, in and around the Toronto area, we are doing this through our Turtle Island Conservation Program by building relationships with First Nations youth to increase awareness and knowledge of species at risk like turtles and bats through outreach and education. And we're doing this through monitoring, research, and sharing of traditional knowledge with elders and knowledge keepers. So Jesse's going to put up a slide which shows some photos of some of this work that we're doing to build relationships with youth to help support local species at risk. So you'll see youth learning about our Blandings Turtle Head Start program, some of the local bat species that First Nations partners are working with us to monitor, and my teammates, Crystal and Ethan, who are leading a lot of this work. They are going out into First Nations and working directly with the youth on workshops and on the land programs. So one program is the Indigenous Youth Guardians Camp and Summit. So in this program, youth learn about the zoo's field conservation programs and the work they do with turtles, snakes, and bats. We also integrate traditional land-based learning through medicine walks, salve making workshops, and storytelling with elders and knowledge sharers. So in the new year, our team will bring the program to First Nations communities as well, working with youth on any of their own projects, like building turtle nest protectors, snake hibernacula, and bat condos. So Jesse, please put up another figure. We're going to talk a little bit about our work with caribou out in the field. So a little further afield, we're working really closely with the First Nations communities on helping to save boreal caribou. Boreal caribou are a really important keystone species whose ecological role is essential to the health and stability of northern boreal ecosystems. So they're a really important cultural species too for many First Nations. They provide a source of food and other materials that are really important to their way of life. Several boreal caribou populations are not self-sustaining in Ontario. That means their numbers are declining rapidly. So they've decreased significantly due to disturbance and degradation of their critical habitat and climate change, which I know we're all learning about a lot now. To safeguard the long-term viability of boreal caribou, we believe that conservation frameworks must go beyond preserving their habitat and should also incorporate some genetic preservation through strategies like cryobanking, research, monitoring in the field, and monitoring. And all of this work is supported by Indigenous knowledge and Indigenous communities. So one community we're partnering with is Weenusk First Nation near the Hudson Bay. So it's over 1,300 kilometers away. So you can see on the map on the slide how far north it is compared to where we are located in Toronto. So some of our work with the First Nation is to collect samples from caribou that the community hunts for food. So in the spring, my colleague, my teammate, and we traveled there to collect samples. And some other teammates, Vanessa and Virginia, just returned from being out in the land with community members. So Jesse, can you please show us some of the photos of Vanessa and Virginia's recent trip? So here are a few images from our trips. They really give you an idea of the beauty of the land and how important the lands and waters are to First Nations people and how we must come together to save these iconic species. On this recent trip, Vanessa and Virginia traveled with community members over 5 hours by snowmobile out on the land to collect caribou samples in minus 30-degree weather. Local guides took really good care of them and showed them how to look for caribou tracks and how weather patterns affect the movement of the animals. We would not be able to do this work without the help and support of the community members. Vanessa and Virginia also gave a presentation at a local school and developed a science assignment so the students can connect their land-based knowledge with Western-based science. So after we collect the samples from the field, what happens next? Well, my colleague Paula is in the lab right now and she'll explain.
Fantastic. Here we go. Welcome in, Paula.
Hi. So thank you so much, Jennifer, for that and for the update on what's going on out in the field. It's absolutely amazing what the crew is doing, what our teams are doing out there. And what happens, as Jennifer says, as it comes to the Toronto Zoo, what then? Well, I have the luxury of staying inside one of the most amazing rooms at the Toronto Zoo, in my opinion, because I am a research scientist. So I am the science development manager here at your Toronto Zoo, which means that I am invested and incorporated into our conservation science program. Now, the Toronto Zoo is a conservation-based organization that runs a world-class zoo. So as conservation the part that I'm most invested in, the part that I work on most, is our reproductive sciences department, or reproductive sciences labs. So here we're looking at ways to help and support animals, especially rare and endangered species, to have babies, so to have offspring. And one of the ways that we can do that is to bank living material, living cells from the animals for the future, long into the future. And how do we do that? Through cryogenics. So cryopreservation of living cells, and in this case for reproductive cells, that would include sperm and eggs, or in science we call them oocytes. We can combine them together, they create an embryo. The embryo are the first few cells of life. So every life started as an embryo that then divides out and becomes who you are. We can freeze those basic cells that start all of that in a cryogenic freezing containment sort of system in our cryogenic lab. So your Toronto Zoo is the only zoo in Canada that has invested in reproductive sciences, and we have Canada's Wildlife Cryobank, where we store hundreds of species of cells. So that would be, again, sperm, eggs, embryos, and also living fibroblasts or somatic cells. This is an example of one of the tanks where these cells would be stored. So what is cryogenics and how does that work? Cryogenics is freezing at well below zero. So maybe on your coldest days, and you heard that our team was out in -30 weather, you might have experienced a -30 day outside in the wintertime. And the ears get really, cold. And depending on where you are in the world, it might have even been a little colder than that. But I promise you, it wasn't as cold as what's in this tank. Inside this tank is liquid nitrogen, and liquid nitrogen in liquid form is -196 degrees. Now, nitrogen is not stable as a liquid. It wants to be a gas. It wants to go into vapor form. So as I open this, you're going to see that it's going to go from liquid and make that transition to vapor as soon as I open this lid. Can you catch that? Did you get that? All right. So oftentimes that's what they say science is, right? It's that smoke and vapor. It's real, it's true, and it's what we see in our lab. So inside this vessel is liquid nitrogen, which is storing and freezing our sperm, eggs, embryos, and fibroblast cells. How do we get them to do that? If I put my hand straight into that liquid nitrogen, I'm going to damage all the cells that it touches because it's way too cold. Just like when you're outside in the cold, you have to bundle up or you're going to hurt your cells. So we have to use very special cryoprotectants in order to do that. Now, a cryoprotectant basically is just a solution that protects the cell and allows it to go to this temperature to get cold enough that it can be stored. When it's at that temperature, there is no enzyme activity, so no protein degradation. It's basically still and static. Nothing is happening. If we've done it correctly, when we thaw that sample, it will still be alive. So we collect a sperm sample from the caribou, taken from the field, contained very carefully with specialized equipment and shipped to us here at the Toronto Zoo. We will remove the sperm from the testes from the animal and cryogenically freeze it. When we thaw that straw, it will be alive and it will be capable of either fertilizing an oocyte in vitro here in our lab or used in artificial insemination to impregnate a female caribou. What is important about that? What's important is that we are now taking genetic material, living material, and extending the amount of time that we can use it. We're capturing very rare genetics, holding on to them so that when we want to repopulate any populations that are crashing, we're not trying to do it from a few select individuals. We're doing it from this larger pool, so larger genetic diversity. Now, diversity is the key to species survival. We have a larger pool with which to draw from to really rebuild that conservation-reliant herd. And we can do that all by storing it here in liquid nitrogen. This is not an easy sort of thing. There are a few industries where this has been applied. So for cattle industry, for the agriculture industry, it's used. Also for human. We have the daunting task of then looking after all the other species, the hundreds of thousands of species that there are after that, to be able to freeze genetic material down. So everything from the caribou that we talked about, all the way down to eastern Massasauga rattlesnakes. Yes, that is true. They're an important part of our ecosystem. So we're trying to figure out how to freeze and maintain genetic diversity in cells from all of these incredibly amazing species here. Now, this is just one example. I'm going to show you what the actual room looks like. We're going to head over here. You're standing in, or I'm standing in and broadcasting to you from our gamete biology lab, where a lot of the magic happens. And these are where all the tanks are stored. So you can see we've got several other tanks similar to the one that I showed you. Now, all of these small tanks are filled with sperm, eggs, and embryos. The larger tank that's back here is filled with somatic cells, so those are the cells from the body. The challenging thing with the animals that we work with is that they're very seasonal, which means that they only produce reproductive cells in a particular time of the year. Now, if they happen to pass away or we have to euthanize for compassionate reasons, we can collect the ovaries and the testes, we can collect the gametes and freeze them, but if they're not in the reproductive season, they're not actually producing any of those gametes. So we then would take cells. We can get a little piece of tissue, we can take the cells out, extract them, and grow them in culture, and then we'll freeze them down. And they are very much alive here in this tank. These tanks stored in here, there's over 100 different species stored in these tanks, and then multiple individuals from those species. This tank over here, I said that nitrogen is not stable, so we have a tank over here that we use to fill our nitrogen tanks at least every week because they're off-gassing, so for us to be able to maintain them, that's what it requires, is that we have liquid nitrogen on site to be able to top up these tanks. we don't actually know the upper limit of cryopreserved cells. They might, the longest case that we've heard of is 80 years. That was in a domestic cow. So from our understanding of basics of cryogenics, this is a lifelong investment. We can actually keep these cells alive for a very long time. It is our hope that we won't have to do this for every species, but as a part of conservation activities, it is a tool that we can use to help and support our wild species and the species that we have in our care from rare and endangered species.
Amazing. Thank you so much. This has been great to see all of our doers here and all of the different cells that we have stored inside. Paula, do we think we're ready to head to our question period?
If you're ready, I'm ready.
We're ready, folks. And I mean, I just want to say the fact that you highlighted this is sort of like the iconic science is like a raising of a thing as like fog pours out. We've done this in a few broadcasts this year, but it's always so fun to go back. And so our first question was about the safety of this. So someone commented that you did that without gloves. Are you safe in being around liquid nitrogen, or are there any precautions you need to take?
That is such a great question, and I'm glad we're in this room to answer it. I'm going to show you what the gloves look like. So, if I was touching the hole, now, I'm going to review what I had done so you can see. This is all at room temperature still. This part and this, room temperature. Had I gone in to grab something out of the tank, I would need gloves. Let's see what that looks like. So these gloves are different than your regular lab gloves, right? You can see the difference of how insulated they are. They are specific for cryogenics. Am I now protected as impermeable to cold? Nope, it will go through. It's just a matter of time. But let me show you. Come a little closer and I'll show you what's inside these tanks. So we have our sperm and cells frozen inside straws. These things that I'm lifting up now are very, very, very cold and actually have a little bit of liquid nitrogen still attached to them. Here's one that has the actual sperm straws you can see there. I'm going to put it back inside so I don't thaw them out. So you're correct. If I was going to reach in and grab something, it would have been cold to the touch. So I do have to wear gloves for that part. Now, if I'm going to be dispensing liquid nitrogen, I also have to wear a face shield. Because the nitrogen, if it gets it into my eye, the drops or any water surfaces, it will freeze it instantly. So part of our protective equipment, personal protective equipment or PPE, is to wear a face mask as well. Great question.
Great question. And by the way, you pulling out the vials, I just instantly started humming the Jurassic Park theme in my head, like the guy taking Dilophosaurus before he goes off. So thank you very much for that in my day. Also, I don't know if they sell those gloves anywhere, but people would buy them in Canada. I can tell you, like that looks fantastic for just a hike around the bush. We're going to head to North Carolina. Lavi, if you want to come on in and take us away with our first live question, you are good to go. Hey.
Hi, how are you doing today?
Nice to see you. I think they're good. They're smiling ear to ear because you're going to ask great questions. That's a good sign.
Yeah. So my first question is, why are you the only ones in Canada who do this? Because it seems like a good way to help species.
Yeah.
Oh, that is such a great question. So it's a bit of a complicated answer. One of the answers that's, sort of front and foremost is that we had the specialized, we had the expertise at hand to set this up. So it just happened that we had somebody here who was capable of doing that. We had somebody who was very interested in endowing the funds for us to be able to do that, so providing the funds for us to be able to set that up. And then we've just been very lucky at being able to hire in talented people to kind of keep the momentum going until it developed into something that became this iconic thing. Now, just because we're the only zoo in Canada that does this type of work doesn't mean that we don't go out and support other zoos. So we support other zoos across Canada. We support other conservation organizations. Whatever needs might be, if we have the expertise to cover them, then we will go out and help and support them. So sometimes conservation dollars are very small and we have to divide them up, and it's like a divide and conquer. So we are specialists at this, and another zoo will be a specialist at something else, and then we can partner in being able to provide a really impactful conservation project or difference in the work that we do by not trying to all of us do the same thing. We lean into our expertise and work together.
So this is the perfect question leading us into the next question, which is, Does the Canadian government or other conservation organizations ask you to store specimens, and do they pay you to do that? So, do tell.
That's a really great question. So actually the Caribou project is a pilot project that has Ontario government funding for us to figure out how to do this. How do we go into communities and how do we collect these samples opportunistically and bank? And so that is a grant funded project. We are speaking with our governments, our federal government level, creating awareness around the benefits of cryobanking and biobanking, and what that could really provide for rare and endangered species. So we're creating awareness and attempting to create a national program for this that would be federal based. Now, this is one part of what reproductive sciences at your Toronto Zoo does. We also do a lot of endocrinology work. Okay, I'll break that down. Endocrinology is the study of hormones. Hormones are very important for reproduction. So we also measure hormones from as many non-invasive samples as possible. What are non-invasive samples? That would be things like poop or urine. We can do hair. That part, we have a lot of support. We support a lot of projects that are government, that are conservation entities, that are zoos, that are consulting firms for wild species all across Canada and actually around the world. So believe it or not, we receive poop in the mail almost weekly, and we extract the hormones and run the hormones. So although the biobank might not necessarily be in full force yet, although we are building it, we're building out to create capacity to be able to support on a national level, we are already in the sphere of national support with our endocrinology work, which is again linked in reproduction. So many ways that we can contribute.
What a great answer. And maybe that explains why I got poop in the mail. They thought I was going to the biobank, but it was meant for you. I'll try and reship it, okay? Good to know. We have so many, the lab coat really appealed to people. We've got some of the best questions I've ever seen in a Q&A in my life, so we're going to take a lot more of those. But Jennifer, I'm going to go to you for a second. So we had a question about how often you do the expeditions out on the land, because so is that every year? Is it multiple times a year? Is there a benchmark for this in any way?
So the Caribou, the work that we were just talking about outside, so going up north and taking samples from the Caribou, that's connected to the project that Paula was just talking about that's funded by the Ontario government. So it really depends on what projects we have going, the funding. So right now we have only gone up to that community, Weenusk First Nation, in the Hudson Bay area twice. And it really depends on the nature of the partnerships that we have, and of course the funding that we get. So we're hoping that we can do this type of work more frequently. But a lot of it really depends on building those really strong relationships with communities and First Nations communities, because when we talk about going out on the land and collecting samples and working with the animals, that means that we are working with the Indigenous communities on whose territory we're on. So we have to be really slow, really respectful, really take the time to build those relationships. Yeah. So that will also dictate how many times we go out on the land and collect samples.
Thank you very much for the nuanced and in-depth answer. And I want to stress for all our audience on the Zoo and Exploring by the Seat of Your Pants YouTube and Facebook channels today, maybe you should encourage people to donate to the zoo so they can do more of these amazing expeditions in science. There we go. So get that funding that we all need. Way to go. That's my plug as a person that's outward from the organization, encouraging you all, because you'll find no better team than the Zoo team. I say that very earnestly. You guys all rock. So, all right. We've got 20 more minutes for questions. This is like an insanely long Q&A, which is great. So thank you very much for doing the deep dive in the beginning. I'm going to take some from our YouTube audience, then we're going to go back to our StreamYard folks. We've got so many questions you can shake a stick at. Elena in grade 2 wants to know how many cells can fit in one tank and how many different types of cells you keep in the lab.
So I was walking us out because there's quite an echo in here, and our fans will turn on at some point. So if you see us run out, we have fans here, and I'm going to answer that question. I just want to address nitrogen. Too much of it will displace oxygen. So we have all of these safety devices in place in order to turn the air over a number of times an hour. We have oxygen sensors that tell us if it's safe to work in here or not. So the fan may kick in on its own because it does that. And when it does, you're not going to be able to hear my melodic tones. So we're going to make a run out of the lab if that happens. But I want to answer these questions first. And I'm going to start with how many samples can we store in a tank? It depends on the type of sample that we're storing. So I'll lift up the cane that's in this tank as well and give you an idea of what this one looks like, because it's a little bit different. So we have our, put the right glove on the right hand. We have our cells here, our somatic cells in this tank, and it looks a little different than what I just showed you. They're stored in what are called cryovials. So they're 2 ml vials that are stored in boxes. So if I pull this cane out, this canister out, you can see we've got boxes of samples that have vials inside of them. So in this tank, we can save 6,000 different vials just in this tank. And we have another tank. This one's actually full. We filled it up this year because of our partnerships with some external, the national growth that I was talking about. We've been receiving some cells from external partners. So part of the Vancouver Island Marmot Recovery Strategy, they're starting to collect tissues for us to grow cells for biobanking. Whooping crane is another one that we've been embedded into their conservation strategy. Sage grouse, another story where we're helping to support again with biobanks. And those samples, those animals are not here, they're being sent to us. Now, we do have some Vancouver Island Marmot here, but these are the ones that are part of the larger sort of recovery program as well. So we've actually met capacity on this one. We have another one coming in. In these tanks, again, it depends on what we're storing. If we're storing straws, we can store just thousands and thousands of straws. If we're storing oocytes or embryos, so the egg or the embryo that's created after the sperm and egg are combined, we can, a little bit less than sperm, but still quite a number in the hundreds to thousands, of those. And then if we're storing cells in here, now we're down to a few hundred because it takes up so much space in these tanks. This is the much more efficient way of storing it. But we have a lot of capacity here to be able to store.
Sweet. As you exit the room so we can hear your melodic tones more beautifully, the quick follow-up on that was how many different types of cells? Is there like 30, 150, 1 million, 5?
That's a big range.
Somewhere between 5 and a million,
Somewhere
I think.
between 5 and a million. So we have predominantly, we collect sperm. Sperm is one of the easiest cells that we can collect on the gamete side. It's a lot easier to collect sperm from the males when they come in, maybe for a procedure, they're under anesthesia. We can opportunistically collect. Again, if our animals pass away or are euthanized for compassionate reasons, we can collect the testes and we can collect the sperm. With our females, it's a bit more challenging. It's much more invasive to be able to get the oocytes from the ovaries. That requires a bit more expertise in a surgical procedure or standing, but more expertise at a veterinary level. And so we have far fewer oocytes, but we do still have some in our tank. We combine those two together. They make embryos. So we're at 3 types now: sperm, oocytes, embryos. Somatic cells, so those are body cells. Those are, again, quite easy for us to collect if we get a piece of tissue. So that's another type. We've recently started collecting for the sage grouse primordial germ cells. So those come from the egg. They're actually a very undeveloped type of cell or undifferentiated type of cell that we've started banking in our cryobank as well. And as we come across more conservation-reliant projects that need us to be more flexible and expand, we will continue to expand out our repertoire of cells. Oh, and embryonic fibroblasts. We also store it at the same time from the sage grouse, embryonic fibroblasts. So what are we at now? 5, 6? Wait, sperm, eggs, embryos, somatic cells, primordial germ cells, embryonic fibroblasts. 6.
Sweet. Never has the number 6 been said more enthusiastically in the history of exploring by the seat of your pants. So thank you for that. We're going to head to Mrs. Davidson's class at the delightfully named Friendship Valley Elementary. Come on in, unmute your mic, and take us away, folks. Hey.
Wait, hold on.
Say hello. Good. Say
Hey. Hi.
We just came in from a fire, Joe. It was really, really cold out there.
Ooh, okay, warm up by the fire, the nice heat in the classroom, and warm up by the fire of these great passionate answers from our speakers.
Are you going to answer your question? Do you remember your question here?
How often do you collect samples?
Nice.
Oh, almost. Definitely weekly, I would say. If we average it out, so if we're talking gamete cells that we're freezing in our biobank, I would say probably if it averaged out to about weekly, we collect samples, and that might not be going out in the wild though. That those samples sometimes are mostly from the animals that are in our care. So we don't
you might notice that I'm a little pale. I tend to be an indoor lab girl. I don't see the sun very often. That's sort of my contribution. I stay inside and I do a lot of the work here. So I don't always go out to the field. And sometimes the field biologists who we partner with, as I said, everybody's got their skill set. They send us the samples, it gets to the lab, and then I do the processing here. So I would average it out to probably weekly, and then some weeks we have just a tremendous number of cells that are coming in. So the Vancouver Island Marmot project that I talked about, they process all of the little pups at the same time, and they took skin samples from them while they were processing them to ship to us. So at any one time we received, I think, 9 to 10 per shipment, and so I think we processed 39 cell tissues into cell cultures. And we had over 100 eggs that came in from the sage grouse. So it really kind of depends on what's going on in that moment and what projects we're supporting. For the caribou, we've got, I think, 3 tissues from 3 individuals that are coming in to the lab. They started the processing when they landed in Timmins, and then they're going to come here to finish and put them in the cryobank.
We admire your long-suffering avoiding of the sun in the pale laboratory that you so clearly love. Well done. Bravo to you on behalf of all of us here as an audience. We're going to do one more quick one from Alla, and then we're going to do one on caribous. We're going to go back to Jennifer in just a minute. But Alla in grade 9, Mrs. Lami's virtual school, and welcome in everybody. They want to know, is there a limit on how long sperm can be frozen? Is there is it a month? Is it a year? A million years? What's the deal?
That is such a great question. And the answer is we don't know. So as far as we're aware, and in my lifetime, it'll still be alive. So we don't know the upper limit to cryopreservation, cryopreserved samples, but it's definitely decades that has been proven. So the oldest known use was the sample had been frozen for 80 years. We here at your Toronto Zoo have thawed sperm that was frozen in our tank for 35 years and did an artificial insemination in our wood bison, and we got a calf. And we still have some of that sperm here. So now it's over 40 years old from the time that we did that. We actually, we don't know an upper limit to this technology. And again, it's as long as we froze it correctly and we maintain these tanks, which is a big part of our job, then there should be no issue, no upper limit to how long we can freeze those cells for and keep those cells, maintain them.
I've got such a cool follow-up question to this from our YouTube audience in just a minute, but we're going to go back to Mrs. Lami's class because they've got to go, and they had an amazing question. I know this is a really big one. We're going to try and keep the answer as short as we can because we want to get in as many as we can. So Shabir wanted to know, what are all the benefits of biobanking and cryogenic research? Like, it seems like there's a lot of cool stuff we can get from this. What would be your go-to answer?
Okay, all the benefits of biobanking. 1, long-term genetic preservation. So we're holding on to genetic diversity and in our tanks and holding it for a long time. So that right away has a benefit. 2, what I didn't show you was, so there's a straw, the sperm straw, the singular straw. We can put it into a smaller tank that can be shipped anywhere in the world. So now I can move sperm from an adult male giraffe and not have to figure out how to move an adult male giraffe. So that is beneficial. It also can help with disease. So a great example is a wood bison. In the wild, approximately half of them are infected with tuberculosis and brucellosis. They're doing an amazing job, the communities up there and also Parks Canada, of keeping those separated. But there is that concern that disease will move. So these animals also are not particularly healthy. So there's an order to have these animals removed from the population so that they don't infect the entire population. We can capture those gametes, we can clean them to remove the tuberculosis and brucellosis, and then they can go into a clean surrogate, and we don't lose that genetic material. If we just cull or harvest all of those animals, remove them from the population, wood bison are threatened, they will become endangered, and we will lose all of that genetic diversity. By biobanking, we can capture, we can freeze, we can hold on to it, and then bring it out later on. It allows us also to be able to match animals. So sometimes breeding is challenging. If you put a male and a female together, that does not mean that they're going to breed and produce offspring. Assisted reproductive technologies based using cryopreserved gametes is a great way to sometimes get around that if we have very, very endangered species and we absolutely need them to breed. So there are a multitude of different things, and there's probably other ones, but I'm trying to be brief.
That's really, really good. Okay. Not only is that like an awesome deep dive on all the benefits of it, and thank you, Shabir, for the awesome question, but I want to stress, you talked about funding earlier, and I'm convinced, and I'm going to pitch this to the whole zoo executive team, that given how your commentary is in this Q&A, if they charge $10 a ticket to watch you try to move an adult male giraffe, you could pay for all the things you want to do for years to come. So please do consider that, Dolph. Please, it would be a lot of fun. All right, Mrs. Davidson's class, we're going to take a question for you guys. Welcome back. Unmute your mic and share away. Hey.
All right, hello. Go ahead and ask your question.
How many caribou do you have at the zoo?
Ooh.
Oh. So I can actually take that question here. I'll hand my stand over here. So hello, everyone. I've been behind the camera here for us. So I believe right now here at the zoo, I believe we have 3 caribou. Herds can change, and our amount of animals that we have physically here, obviously we have our preserved animals in our dewars, but then also our physical animals that we have here can change a little bit as well. And we've talked about this before on broadcast, but how we get animals here. And so long times ago, zoos were taking animals from the wild, which we do not do anymore. That is not the goal of it. As Paula was saying, we are a conservation facility with an outstanding zoo attached to it here, or kind of in combination with it. So we want to make sure that the animals that we have here have purpose, animals live with purpose, and they are helping to be ambassadors and educational ambassadors for their species and for the conservation work that these amazing teams are doing here as well. So right now, I believe we have a herd of 3 caribou. That number can change, though. If another zoo has caribou that they need to bring here, they might come here. If ours are older and maybe pass away, we might seek out other ones to, again, be able to have that ambassador species, promote and help to connect those of you who are on the ground, maybe growing up wanting to become researchers or educators yourselves. It really helps you build that connection with our species as well. And I think Paula has something to add to it here as well.
I have one caveat to that explanation from Mary Ellen, is that there are some very rare times when we do actually step in and take animals from the wild, and I'm going to explain very clearly why that is. You may not know this, but your Toronto Zoo and several other zoos around the world, accredited zoos, actually do conservation breeding and release. So we breed animals here and we release them back into the wild. The reason that we're doing that is because in the wild, the population numbers were just decimated down to almost nothing. And if we hadn't stepped in and brought them into a safe space for breeding and reintroduction, they would most certainly be extinct. So we are part of a number of breeding and reintroduction programs. Vancouver Island Marmot is one, and they're doing an amazing job repopulating the species out in Vancouver. That is Canada's most endangered mammal. Eastern Massasauga rattlesnake, we had up until very recently for about 4 decades participated in the black-footed ferret breeding and release program. But although we are closing that program, it's not that we're not interested or invested in it. It's that we're redirecting it to support a lot of Canadian species that we're working on. Eastern loggerhead shrike, yet another example. So many examples of how we step in and do that. So a little caveat to that. Sometimes we do have to do that, but the idea is to support the wild populations for reintroduction. So
thank you again for the in-depth answer, guys. This has been such a great Q&A, man. Okay, I'm going to take an amazing one from YouTube, and please, Miss Claremont, we want your remaining questions. Anyone else who has extra questions that you don't get answered in the chat today, please email them to us at exploringbytheseat of If you registered, we'll pass them along to the zoo team and inundate them with great questions. This is such a neat follow-up. How does this process, when you're taking these samples, account for any evolution the species might be undergoing in the natural environment? If you have samples for 40 years, the species change over time. It's a crazy question. So well done to St. Carlo Virtual School for that. No pressure.
wow. You're not making it easy on me, guys. This is some big, heavy hitting stuff. I'm going to start with evolution. Typically is a very slow process that happens over just, a very long time. Unless you are born and dying, within a very short window. So like fruit flies. So fruit flies are a good example of a model species that we do a lot of genetics on, because their life cycle is very short. And you can actually make changes, phenotypically observable changes in the Drosophila and fruit flies really quickly, and be able to see and see what's happening with the genetic patterns or what's going on when you make these manipulations or these breeding sort of changes. But with mammals and a lot of other species, and reptiles in particular, are incredibly slow. You might think that they look prehistoric because they do. They evolve very slowly. Some of these animals, the evolution is so slow that what we hold on to in our tanks is unlikely to have any real impact on the evolution of the animal itself. Now, what we might see, though, are epigenetic changes. So evolutionary genetic changes are about the genes themselves, the whole genome in that animal. Epigenetic changes are how it's folded, more or less. That's a very simplistic way of looking at it, but how it's folded. And that folding and conformation can be impacted by environmental things that could potentially be an issue with maybe handling and cryogenic freezing. The epigenetic changes tend not to be large enough to cause any real impact. However, the reason that we don't really monitor epigenetic changes with our animals is because we're talking about rare and endangered species. Epigenetic concerns are really for a very rich population where you've got lots and lots and lots of individuals, and now you're just getting down to the minutia of the errors. When we're talking about 10 animals that we're trying to rebuild an entire population from, and that's not an exaggeration, black-footed ferrets, there were 18 individuals brought in from the wild that have built the entire conservation breeding reintroduction program of all the ferrets that are out there now. Epigenetic changes are not a concern for us. We just need feet on the ground. So basic genetics. We even know that there could be genetic issues there as well. That's an amazing question, and I'm not sure if I fully answered it because I think partly cryogenics hasn't been around long enough for us to know what the genetic impacts could be. We're talking like thousands of years yet to go. I'd love to be around. Or would I love to be around 1,000 years?
Would you love to be around? That's our next question. Do you want to live forever?
That is, but yeah. At the moment we don't know that would have that kind of impact.
Please do. Honestly, audience, again, there's more questions that we can possibly take in one Q&A. You can email us at this email address. We'd love to share them with the zoo team and inundate their inboxes when we're done. I want to stress, too, because you've mentioned black-footed ferrets a couple times, on our YouTube channel, Christy Bly from World Wildlife Fund has some amazing black-footed ferret programs, so you can explore and discover with them, too. Paula, we're going to wrap up with one final question from Levi before we say goodbye. Come on in North Carolina and take us away. Hey.
Hi. So my last question was, you spoke about measuring hormones. Do you measure proteins?
Ooh.
Oh, wow. My gosh, you guys are great. That's true. You guys brought your science A game to these questions. This is amazing. So actually some hormones are protein-based hormones. So now we're talking about things like luteinizing hormone and follicle stimulating hormone. Yes, we can, but not in feces. In those particular instances, we'd be measuring them in blood samples, so serum or plasma samples. We can actually measure those types of hormones. Now, proteins as a basis, technically, yes, and as a science sort of entity or the sciences of reproductive science, yeah, understanding proteins in a body and how that impacts reproduction is a really important part of science. Here at the Toronto Zoo, we don't really do a lot of that type of work. We're not really set up for Western blots, but we can, or mass spec, but I'm now throwing out a lot of terms you're going to have to look up. As my parting gift to you, a little bit of homework. But yeah, proteins are an important part that we have incorporated into some of our projects. Not one that we heavily lean into though, but we do have partners here in Toronto, institutions and facilities that we can work with to do some of that work. But yeah, amazing question. Excellent question. Proteins are incredibly important, of course. Yeah.
We could take questions all day long, and we literally have enough to do that. But I know time flies and you're having fun. We are nearing the end of the broadcast. So before we wrap up this program and this amazing series in collaboration with the zoo, is there any final message on both sides from both of you that you'd like to share with us before we say goodbye and bring in our classes for a farewell?
Well, one final message I would like to say is how important people are and communities are to conservation and to saving species. So we all have a role to play. We're not separate from nature. We are part of it. So just remember that when you're out in your communities, when you're out in the land, that you have a role to play. And yeah, that's my parting words.
And my parting words to build on what Jennifer has said are the partnerships that we build and the our ability to share our messaging. And so our important work with Jennifer, we cannot do this alone. No zoo, no lab, myself, none of us, we do this alone. We work in community together. We work in partnership together to accomplish these things, to accomplish these amazing goals. And it isn't just because I'm the one wearing the lab coat and I'm standing here in this lab doesn't mean that you guys aren't actually incredibly important in the success of zoos and conservation programs to be able to save these rare and endangered species that we're so incredibly passionate about saving.
What a great final message from the both of you. Thank you so much for your passion, your expertise, all of it. Check out torontozoo.com, everyone. Check out the Amazing Guardian site. All these are on our YouTube channel in a special playlist already. So it's been such a thrill having you on for the last 5 weeks. And a big Merry Christmas, Happy Holidays to everyone. As we do to wrap up every series and program, I'm going to bring in all our classes to say a big thank you and farewell. So la vie, Mrs. Davidson's class, everyone at home on YouTube, thank you so much.
Thank you.